Welcome, everyone. It's a great pleasure to talk about Nanovation and our efforts. And there couldn't be, I think, a better time after or following our announcement of the big partnership with Novo Nordisk from last week to talk about extrahepatic delivery. And Peter already gave a great segue into this topic, which is really the breakthroughs we have seen for delivering RNA to the liver for ONPETRO or the COVID-19 vaccine. So there's such a massive potential now for genomic medicines, and there are various different organs, obviously, in our body, from bone marrow to the immune system, to liver, to local administration, to the eye, central nervous systems. And as Peter alluded to, there are many, many different clinical trials for various different diseases like infectious diseases, oncology, metabolic diseases, hepatology. So there's such a big potential, but it really comes down to one point, which is delivery. And how can we overcome this delivery barrier and really access all these different tissues which go beyond the liver? And this was really the focus of this talk and providing a bit of background. And there's obviously a lot we can talk about what we have done as a company and the entire team of Nanovation. And I will provide an overview and will be more than happy to provide any answers at the Q&A, or then you can more than welcome to contact us. So before talking about Nanovation, just want to highlight what Peter had said with ONPETRO. We have the typical ONPETRO composition, which is 50% ionizable lipids, 10% phospholipid, 38.5 mole percent of cholesterol, 1.5% of a diffusible PEG lipid. Injected IV, all these LNPs go straight to the liver. It's a perfect tool to engage with RNA delivery effectively to the liver and therefore treat diseases by gene silencing or gene expression. The question is now, how do you get access to other tissues? And I have a short question to all of you, and Alex, I asked you to start the short poll. In terms of what is the circulation half-life of ONPETRO in mice? So if you inject an ONPETRO formulation in mice, how quickly is it eliminated? If you can vote for one of these answers, 15 to 30 minutes, 6 to 8 hours, 20 to 24 hours. So I see that roughly 60%, 2/3 of the crowd says 15 to 30 minutes, but the other 40 to 50% says 6 to 8, 20 to 24 hours. And the answer is the following, that the elimination of ONPETRO of a liver-tropic LNP is very quick. You see here the percent injected dose in blood over time. So the elimination is very quick. The half-life is 15 to 30 minutes. There's a slight dose dependency, but this enables a really quick accumulation in the liver and therefore very effective targeting. But what you can imagine from this, it's pretty difficult to target extrahepatic tissues if it's eliminated so quickly from the blood and goes straight into the liver as well as into the spleen. So what have been approaches others have used in the past? And there are two main approaches. One is we have the diffusible PEG lipids on the LNP. So you can either up the dose of the PEG lipid and therefore increase the circulation half-life in blood, or you can say you have a non-diffusible PEG, like the PEG C18 lipid, which anchors really well in the LNP and therefore leads to an increase in circulation half-life. The problem with these two approaches is twofold. One is you run into the so-called PEG dilemma, that the more PEG you put on the surface, the more you limit the transfection competency of an LNP. And if you use non-diffusible PEG, like the PEG C18, like DSG, there's much more adaptive immune responses which prevent potentially repeat dosing. So how do we tackle that problem, that we need a longer circulation half-life in blood to target extrahepatic tissues, but at the same time we need to be transfection potent? And this is really where this concept of Nanovation comes into play. There is no one fits all, and I think our community now realizes this, that you cannot use one LNP for all the different applications. And when we think about extrahepatic delivery, it really comes down to systemic circulation as a major barrier. They need to be transfection potent, as well as they need a high therapeutic index. And Peter alluded to this, that a LNP needs to be very safe as well as effective to make it into the clinic. So what have we done at Nanovation? When we started Nanovation, the goal was really to build a team with experts in the field of lipid chemistry, including Marco, LNP delivery, as well as RNA design, to really build a portfolio that we can empower our partners with a unique toolbox to develop these fit-for-purpose LNPs. And then we'll explain in the second half of my talk how we rationally designed the long circulating LNP, which enables us to target extrahepatic tissues. So it all comes down to rational design of LNPs and understanding what the application of the LNP is. And you hopefully through my talk, you will understand the importance of that aspect. So what we have done at Nanovation is building what we call a one-stop shop IP portfolio. So our IP portfolio covers specialty lipids, including ionizable lipids, with a focus on higher therapeutic index, different LNP compositions, and I will today focus on extrahepatic delivery, and then surface modification to improve tolerability or targeting. And also the payload plays obviously a crucial role, and we will see some of that aspect in Laura's talk, to increase translation or also specificity. So I won't allude too much on the fourth point, but it's a really critical point in the design and development of a lipid nanoparticle RNA drug. And how do we work with partners before jumping into the topic of the long circulating LNP? It's all based on the IP portfolio and the technologies we have developed, which enables us to build these fit for purpose LNPs for different applications like systemic administration, local administration, or ex vivo, and then to enable our partner a clear path into the clinic and an option to license the entire toolbox, which encompasses all the different pieces I have mentioned. So what is the different strategy to target extrahepatic tissues? You can obviously use an ex vivo approach, and you can modify T cells, and Laura will talk about this, using not only viral vectors, but you can actually use LNPs, and we have quite some strong technologies on that front. You can use local administration, what we have seen intramuscular injection for the COVID-19 vaccines, but you can use the same approach, for example, intratumoral injection or intracardial injection or injection into the eye. And there's the systemic approach, like the intravenous administration, and therefore targeting inside the body the different extrahepatic tissues. And today I will only focus on the technology we have developed for systemic administration, which is the so-called long circulating LNP technology. There are some technologies and applications I will mention, and I was just curious, which extrahepatic targets do you find most interesting? Alex, can you do me a favor and start with Paul? And these are obviously some cell types which go beyond the hepatocytes, which is the most common, let's say, low-hanging fruit. Thanks a lot for your participation. It seems T cells is like 36%, hematopoietic stem cells and macrophages, monocytes 25%, dendritic cells 22%. All of these cell types obviously have a really crucial implication for, let's say, rare diseases, for HSCs, T cells, for cancer therapy, or other rare diseases and inflammatory diseases, dendritic cells for cancer vaccines, max monos, for example, for core M therapy. So there's a clear favorite target cell type from the audience, and this also shows how important it is to consider the different cell types we need to target. So jumping right into the extrahepatic delivery and the rationally designed long circulating LNP technology, the LC-LNP. So it really goes back to the point of Peter's career. Like from 1961, the development of liposomes had started and then culminated in the development of Onpattro as a lipid nanoparticle. But what we have seen there, that it went from a liposomal drug like Doxil, the liposomal doxorubicin, which had extended circulation lifetimes of blood, and therefore enabled the accumulation in tumors like the Kaposi sarcoma in this hallmark paper from Harrington et al. Or then we have the development of Markibo, which is a sphingomyelin cholesterol formulation without a PEG lipid, which enables a significantly higher total plasma vincristine concentration. Here you see the liposomal formulation versus the free drug. So by encapsulating a small drug into a lipid bilayer containing nanomedicine, you can increase the circulation half-life. And we took a similar approach to understand how can we design a system which isn't this solid core structure, which has all the different components on the surface, but how can we design a more liposomal-like structure and morphology? And this was really when we started from a conventional LNP through iterative optimization and redesign to come to the long circulating LNP, which is more this bright X structure, which has a lipid bilayer and a distinct core, as Peter has alluded to. And how does this work in a simple way? And there's obviously a lot of variables which come into play. It's the formulation method, the formulation buffer, it's the different helper lipids, it's the different ionizable lipids. But the principle is that when we go from a on-petro-like formulation with 10% of helper lipids and increase this into, like, for example, 50 more percent, you see that there's a key change in the morphology, which then leads to these bilayer structures and the liposomal-like LNPs. And this really enables a different pharmacokinetic profile. So when we inject an LNP compared to a conventional 4-component 50-10, 38.5, 1.5 mole percent composition, and you look at the SPECT-CT after 8 hours in mice, you see a clear, distinct differentiation. On-petro accumulates very strongly in the liver, whereas a long circulating LNP enhances the circulation half-life from 30 minutes for on-petro or 15 minutes to several hours, and you can fine-tune this by the lipid composition. And therefore, we enable the LNP to actually access extrahepatic tissues. So we really have, in the first step, overcame this problem of systemic circulation. The second problem or challenge, which I mentioned, is the therapeutic index. So a simple explanation here, when you inject an on-petro-like composition, which goes straight into the liver, and LNP composition at a single dose of 5 mg of mRNA per kilogram of animal weight, you obviously have really high ALT, AST levels, like liver biomarkers, which are incredibly high for a liver tropic formulation. When you detarget the liver, obviously you lose this toxicity and you have a much better safety profile. And this also is the case for repeated administration. So when we do a repeat administration of an LNP, we don't see an increase in the ALT and AST level. No elevation in cytokines, and we have tested this in mice as well as NHP, and also at different higher doses and repeated administration. So the second challenge following the systemic circulation time is the safety profile. And then the third one is the transfection potency. When we look at the most important component of an LNP, it's the ionizable lipid. So what typically is done is taking different ionizable lipids, putting them in a LNP, which goes to the liver, use an mRNA encoding for luciferase, inject this into mice, and then look at the luciferase expression in the liver. So we have designed various different lipids and screened them more than 250 in vivo. And what you see is when you take MC3, which is the ionizable lipid in the on-petro formulation, or you take ALC 0315 from Comirnaty, there's an increase in efficacy when you go from MC3 to ALC. We have developed lipids which are equally potent or more potent than these clinically approved drugs. This is when it comes to an LNP, which is designed to go to the liver. But how does this look like now for an LNP? And I would like you to focus on the left-hand graph. So if we take now the same ionizable lipid, MC3, package it into an on-petro formulation, or package MC3 as an ionizable lipid in an LNP, you see that we get a significantly higher expression of a reporter gene in the bone marrow, because the LNP has a much higher accumulation and is transfection potent, and therefore you can significantly increase the transfection rate, in this case in the bone marrow. When we combine this now with our lipid library, which is the beauty of having a chemistry team, designing lipids, formulation team, RNA team, preclinical team in-house, is that we can further boost this expression by several fold compared to the MC3 or ALC compositions in a long circulating LNP. So this really enables us to target the bone marrow and different cell types within the bone marrow in a project or in a partnership focused on that tissue. And I just want to give 2 examples here of cell types you can target in the bone marrow. Is hematopoietic stem cells, as we alluded to at the start. This is one of the very important cell types or where many rare diseases originate from. So we could show that when we take an LNP and benchmark this to a standard clinically approved formulation, that we can significantly increase the transfection of the HSCs in a humanized mouse model. The same applies when we look at T cells in the bone marrow. Obviously, T cells also reside in different other tissues, so we'll see some of that when you talk to us. So this is a significant improvement which we have made to increase circulation half-life, increase the therapeutic index, and also keep up with the efficacy and enable relevant doses in the target tissues. So to summarize that part, it's really about how did we overcome the challenge of extrahepatic delivery which others face while targeting the liver. When you take a standard LNP, there's a quick elimination, as we have discussed at the beginning, and it quickly goes to the liver. So it's really tough to target extrahepatic tissues. There is the liver toxicity, which is typically seen for liver tropic formulation. And there's the potency issue if you take approaches like highly stable PEG on the surface of an LNP, that you run into this PEG dilemma. With an LNP, you have increased circulation lifetimes, you have the therapeutic index, and the beauty of working with Nanovation is a great IP position to leverage this technology for different applications, which could be also, and there might be some questions we have seen during the registration, if you take a targeting ligand to modify an LNP, it's clear that the LNP is a much better base formulation for such an approach as compared to a formulation which goes straight into the liver. And with this, I want to conclude in my next 4 slides that nucleic acid therapeutics, and this is my strong personal opinion, are limitless. We live in such a great and historical moment of human, like mankind, and like the medical revolution is really there, because we can now really not only treat, but really cure and prevent diseases using nucleic acid therapeutics. But it comes all down to delivery, which has its limitations. So what we have seen during COVID was obviously with Onpattro, approved in 2018, and then like having a market of like 500 million. COVID-19 vaccines in 22 of 32 billion. There was a nice statement during COVID from Ugur Sahin, BioNTech CEO, who said like 30% of all drugs in the future will be nucleic acid based. Obviously, we see that currently there's a challenge for tackling various different tissues. And to really make this happen, we really have to understand that we need different LNPs for different applications. We have really nice data on ex vivo application and using specifically designed LNPs for such a purpose, for knockout and knock-in of genes into primary T cells. Local administration, the beauty of a intramuscularly injected LNP RNA vaccine, we have various efforts with different partners, or also, as said, like intratumoral injection. LNPs have a lot of like potential. And third, and one of the most hot topics, I would say, is systemic administration of an LNP to target extrahepatic tissues and cell types. So I hope it was clear that we really need this fit-for-purpose development, which is based on the target product profile. You need to understand what is the target tissue, what is the route of administration, what is the payload? Is it an RNA? Is it a guide RNA plus mRNA? Is it a DNA? How to modify it? And what is the application? Do you need a hit and run approach, like for gene editing? Go in, go out as quickly as possible? Or do you need a long-term expression if you want to do protein replacement therapy? And with this in mind, we typically work with partners and closely understand what the target product profile is and how to design the LNP based on that. And with this is my last question in the last call. Who believes that delivery technologies are key to enabling genetic medicine revolution? Okay, there's 100%. So I achieved what I wanted to, and really, I think it's such an incredible time for us as a community, especially for large RNAs, but even for the siRNAs. There's no way around the delivery technology to really break this in vivo barrier. And with that, thanks a lot, ETPN, for helping to organize this webinar. I welcome any question, and if you want to know more about all the different technologies, please contact us. And with that, a big thank you to the entire team. All of this obviously is a lot of work from the entire team at Nanovation, and I'm really proud to be the CEO of such an incredible team.